Good morning, everybody. I hope you all are staying safe and healthy during quarantine time, and I hope you guys are all keeping yourself occupied during our enforced break. Today, we are going to go over a prokaryotic replication fork. Now, here we have a prokaryotic replication fork. Now, as you can see, we've got a lot of things going on here, so I'm going to break them down one by one. First up, we have a gyrase. Now, normally, DNA is tightly bound in its double helix spiral format. What a gyrase does is it goes through and it untwists the DNA and flattens it out, and that makes it a little bit easier for us to commence with our replication. Now, replication begins at a place that's not actually displayed on this diagram right here, but it starts at a place called the origin of replication. Now, that's not too heavy a concept. All it is that it's a sequence in a genome where replication is prescribed to begin. Now, from there enters one enzyme called DNA helicase. What helicase does is it goes through and it unzips our now flat DNA into two strands called the lagging strand and the leading strand. Now, you might wonder how DNA, especially being such an almost fragile molecule, how it stays compact when it's being split apart by DNA helicase, and that's because there are bonds in the backbone of this DNA called phosphodiester bonds. Now, what these are is these are formed between the 3' and the 5' carbons in the backbone of the DNA, and that's what keeps the DNA helps it maintain its structural integrity, so it doesn't break apart and nothing happens to it during this process. Now that DNA is split into the lagging strand and the leading strand, what that does is it makes it a little bit easier for us and for all these enzymes and proteins to go about doing their job and commence with DNA replication. And we're going to go through these enzymes one by one, starting off with primase. Now, primase serves a very important function, and I don't particularly like how it's displayed on this diagram, but this arrow might be able to help you out, showing that primase is moving to the right from 5' to 3' along the lagging strand, and what it does is it lays down one single nucleotide called the RNA primer. Now, what this does is it just signals that DNA synthesis needs to occur here. Now, DNA synthesis itself is done by another enzyme. This is called DNA polymerase. Now, there are two DNA polymerases, and we'll get into the second one in a minute. But DNA polymerase 1 is the one that works on the lagging strand. Now, as it moves along the lagging strand, it moves in the 5' to the 3' direction. And what it does is when this DNA is being replicated, it's replicated in segments called Okazaki fragments. What DNA polymerase does is it goes through and it connects the Okazaki fragments back to the lagging strand. And that process itself is called DNA synthesis, when you bring those two nucleotides together. Now, however, DNA polymerase isn't exactly infallible. The moment where it begins, it actually is unable, and also as it moves through, DNA polymerase connects the Okazaki fragments, and then it removes the RNA primer that our primase has already left behind and replaces it with a DNA nucleotide, and thus completing its segment of DNA synthesis. Now, however, it's not infallible. DNA polymerase works after it's already begun, if that makes sense. What it does is the moment that it begins, it actually leaves a nick behind. It leaves a segment where there is no nucleotide. And now that leaves it up to this other protein called DNA ligase to follow behind it and make sure that it takes up and it takes care of all the nicks and inserts nucleotides where they need be. Now, moving on to our second type of DNA polymerase, and that is called DNA polymerase III. Now, DNA polymerase III works on the leading strand, and some might say it works a little bit better even than DNA polymerase I because it functions without leaving behind any nicks and without having to mess around with Okazaki fragments and all that. It's much more simple. It moves on the leading strand in the 3' to 5' direction, and it doesn't really require any sort of ligase working in behind it. Now, last but not least, we have these proteins right here called single-stranded binding proteins. Now, what they do in prokaryotes is, as well as stabilize, they also modulate all these proteins and enzymes needed for replication and make sure that we're doing our job properly, everything is staying good. Alright guys, thank you guys so much for watching. Goodbye.